As Paul mentioned, now I am coordinating the neurorehabilitation group, and well, basically, our goals are to both understand and provide means by technology to control the human biological system, with more emphasis on the motor function and their interaction with the environment, aiming at improving functional and health outcomes of patients that are affected by neuromotor disorders such as stroke, spinal cord injury, cerebral palsy, Parkinson's disease. And we try to do this with a multidisciplinary methodology that combines mostly robotics, functional electrical stimulation, control approaches, and biomechanics and modeling. So we are in the Cajal Institute in Madrid, which is a very large neuroscience institution that is mostly devoted to basic science, and we are part of the translational neuroscience department. So we do more macroscopic scale type of research, and we do a lot of applied research with humans. And our main expertise and the leaders, what we are the leaders of is wearable robotics in general. So we have been proposing and delivering a number of generations of exoskeletons for walking mostly, and the interfaces to connect patients that may use this type of technologies for training and for rehabilitation. So we also have lots of projects on neuromodulation, brain-computer interfaces, the use of biofeedback, which I will discuss in this presentation as well, and so on. And we also have another research line on more focus on neurorobotics and how we can use biology as inspiration to design, for instance, biped locomotion, and how we can use these robotic platforms as sort of models to understand how we humans control the movement and how we solve the interactions of our segments with the environment. So the outline of the presentation is the following. I will focus mostly today on stroke rehabilitation. So the prior presentation is perfect and has been perfect to introduce a lot of the ideas and topics that I will discuss. So thank you for that, because you have prepared very well the audience for what I will be presenting. I will focus my presentation on gait, so how stroke affects human locomotion, and then revise some of the strategies that you could use both with traditional intervention but also with robotic devices to try to promote recovery and induce motor learning that could be beneficial for neuroplasticity and in turn for recovery of function. So I have designed the presentation in two main parts. The first one will be a presentation of part of the results that we have recently published on a longitudinal study in which we analyze what would happen if you change the biofeedback content while delivering robotic gait training in subacute stroke patients. In the second part, I will discuss an even more recent approach that we are working on at the moment, and some preliminary results that are still unpublished but quite promising and interesting for us, in which we show how we can build control strategies at the robotic side to try to train the control or retrain the control of ankle muscles also in stroke. And I will try to wrap up with some of the main conclusions. So we have already seen that stroke is unfortunately highly prevalent in our societies. There are more than 30 million strokes per year, according to recent data. And as we have seen, there are two main types of stroke, either ischemic or hemorrhagic. The ischemic occurs when, as you have seen, a blood vessel supplying blood to the brain is obstructed, and this leads to brain death or a failure of the functioning of the specific networks in the brain. This accounts for more than the 80% of strokes. And on the other hand, you have hemorrhagic stroke, which is when a blood vessel ruptures, and the most common cause of hemorrhagic stroke is some uncontrolled blood pressure. And this damage is basically like a poison in these areas of the brain that leads to death or damage of the cells and can result in symptoms associated to the pathology. So what are the symptoms? Well, the symptoms are not only individual features that you could isolatedly treat, but this is just to remind that there are a lot of things that go wrong when a patient suffers a stroke. So for instance, as we have seen before, there are problems in producing words, in talking, some cognitive dysfunction related to the attention, to memory, and so on. On the motor control side, of course, the patients may suffer from abnormal motor control patterns of the limbs that affect, in particular, the gait and balance. And a very crucial problem for patients in their daily life is spasticity that, for example, can affect the reaching tasks, but it can also affect the control of the ankle joint, for instance, leading to high levels of problems for independent living, and so on. So, ideally, all those main symptoms should be treated if we want to really help the patients. But of course, with robotic technologies or with neural feedback or functional connectivity metrics, you have to focus where you want to try to help. So we have to try to combine tools. So in summary, what happens for walking in human walking after a stroke? So the function of the cerebral cortex becomes somehow impaired depending on the location of the lesion and the size of the lesion, while the spinal cord is, of course, preserved. There might be some kind of reorganization also at the spinal cord level, but the main idea is that the descending input that is critical for functional walking should be restored or compensated for in some way if we want to induce some long-lasting changes in this type of patients. So one of the types of strokes leads to hemiparesis, which is defined as a muscular weakness or paralysis of a side of the body. And this results in some functional deficits in walking. As we can see here, this patient, for instance, has an stereotyped type of abnormal walking. If you look at the affected side, the more common feature is that you have a longer than normal gait cycle duration. So it takes longer for the affected side to achieve the next step, while the relationship between the stance time and the swing time is lower than normal. And the contrary happens on the right side. So these are what the type of characteristics that you observe externally or with video analysis or even with motion analysis. And there is another associated problem that I have mentioned, that is spasticity, which is a motor disorder that is characterized by velocity-dependent hyperexcitability in the stretch reflexes. So basically, there is an enhanced response to a stretch reflex. And this is a very critical problem for patients. So it's not only about the spasms, but it is also related to abnormal contraction of the flexor and extensor muscles around the joint. So all these things have to be taken into account when you want to try to recover gait and induce motor learning in stroke patients. So, as we have seen, there are asymmetrical deficits that could be pronounced due to hemiparesis. And moreover, there are complex interactions of the neuromusculoskeletal system that should be considered when selecting a treatment method that should act not only at the observable features of this asymmetry, but should act on the underlying pathomechanisms that are causing the disturbances. So this is a big statement and a complicated scenario. So this led us to point out the question. Should the functional recovery be the primary target of standardized rehabilitation, or should we try to focus our attention on the neural mechanisms that should support the recovery? In other words, how shall we target practice-induced motor skill training? Ultimately, what we want is the patients to relearn this motor skill, to improve progressively, but it's not very clear how shall we target the rehabilitation treatment, at least for walking. Why is that? Because you may see improvements, for instance, in symmetry, but these are not necessarily correlated to improvements in the EMG signals, in the coordination patterns of the compensatory mechanisms that explain this improvement in symmetry. And this has been reported in the literature by, for instance, the studies by Jaap Burke in the Netherlands, and so on. So it's not really clear how shall we target this type of gait rehabilitation interventions. And now I will move to analyze which are the main motor learning principles that affect at the central nervous system level when after a stroke, and how could we theoretically take into account these motor learning principles. So first, there is an interplay between the plasticity that could be induced with a user-dependent activity or with some specific training of specific tasks that could lead to physiological changes, but it could also lead to neuroanatomic changes. Okay, so there are two possible effects of this type of plasticity, and this is all integrated under the scheme of what is known as motor learning. So if you have activity-dependent plasticity that could be induced by, for instance, a specific training protocol, this could lead to both anatomical or physiological changes. So anatomical level, one example has been demonstrated in a study with an animal model in which an animal model of stroke, in which Nudo and colleagues, they have shown that the cortical representation of the leg muscles in the rat can be changed once the rat is retrained to improve the skill. And conversely, this has been at the neurophysiological level demonstrated by the work of Monica Perez and Nielsen, in which they have shown that using motor evoke potentials, as has been previously introduced, it is possible to induce skill training and in turn changing the excitability of the leg cortical area in healthy humans. And we are using this type of markers, using TMS and analyzing the motor evoke potentials of specific muscles at the lower limb in our protocols and with our robotic interventions. And we'll come back to this later. But in summary, the point is that these two effects are not necessarily correlated, and it's not very clear how they relate, but some scientists have created a line between these two type of demonstrations or observations. So with regard to the recovery after stroke and the strategies that could be used for motor skill acquisition in motor tasks, there is one key theory that is called the challenge point theory that establishes that the outcomes of the rehabilitation could be maximized when the effort is adequate but it's not excessive. So you should try to demand the effort in a way that it doesn't go beyond an optimal challenge point. So here we see that if you have learning at the beginning, if you have low difficulty, your performance can increase progressively due to learning, and until it reaches some challenge point. But after this, the difficulty will start to decrease. So theoretically, you should try to induce or apply this type of difficulty as a function of the performance. And we are using this concept in our development, and I will explain a little bit more later. But some other researchers have, of course, used the general concept to try to develop different types of robotic assistive controllers. So I will explain three of the different types of available controllers that have been tested both for upper limb rehabilitation and lower limb in the next slides. So the first one is called robotic guidance. So basically, what this proposes is that there is a system that is externally supervising how the patient is following a predefined trajectory, and there is a kind of a tunnel of allowed deviation around this prescribed trajectory. So you are pushing the user or the patient towards this predefined trajectory, hoping that this could help to this motor skill acquisition. In general, this has shown to fail massively in what has been explored in the literature for both upper limb rehabilitation with powered exoskeletons, but also with lower limb exoskeletons. So robotic guidance is not good enough, in our opinion. The other demonstration that we have seen is what is called random disturbances, in which improved motor learning has been demonstrated when the patient is executing a task and then suddenly receives a perturbation randomly in time. So the idea behind this, or underlying this concept, is that this variability introduced to the task may increase the recovery because the patient is more motivated to have higher attention and adherence to the realization of the task. The problem, in my opinion, with this type of randomness, temporal randomness, is that it's excluding the idea of having some kind of prediction of what's going to happen that should be considered for the creation of an internal model that, in theory, should account for the response to the perturbations coming from the environment. So this idea is interesting, and it has shown to be useful, at least for healthy users performing dorsiplantar flexion tasks to induce motor learning, but it's not yet clear to what degree is something that could be transferred to patients. This is the work mostly by Marchand Crespo in Switzerland. And the other approach, which is much more appealing and perhaps more related to this challenge point theory, is called error augmentation approach, which basically has been proposed to enhance motor learning, so that, for instance, the patient ideally should try to follow this trajectory, but at the beginning they are not able to do so. So what you are doing is to perturb them outside of this trajectory, so the contrary to robotic guidance. So you are providing these forces, trying to deviate the user from this trajectory, and then if you remove these forces, you expect that this will induce a skill acquisition. So Reikens-Meyer and Patton have suggested that starting with guidance first could be relevant at the beginning, but then shifting to this type of error augmentation could be more useful for patients to lead to motor learning. On the other hand, this type of interventions at joint level, at individual joint level, could be informed by visual feedback to the users. So these are examples of robotic or actuated systems at the ankle joint, and different types of visual feedback have been used in order to, for instance, apply this kind of random disturbances while the patient is trying to control the ankle joint, for instance, to move around a helicopter, or so on. So this is just to show that visual feedback is very useful in this type of motor rehabilitation, with some sort of haptic feedback to challenge the patient. On the other hand, well, there are different types of wearable exoskeletons for gait training. So there are two main classes, if we want to make a quick classification. Those that are based on body weight support systems, that are useful for patients that are heavily affected. So basically, this type of exoskeletons are guiding the joints externally, trying to replicate a normal motor pattern, and they are applying the robotic guidance approach that I have explained before. On the other hand, there are these modular exoskeletons that could be used to actuate joints independently, or more sophisticated 6 degrees of freedom overground exoskeletons to try to do something similar as the Locomat, but on the ground. So if we concentrate on what has been demonstrated with regard to the use of the Locomat system, which is this device, by the way, for training stroke patients, if compared to standard rehabilitation with regard to spasticity, there are no demonstrated results about the efficacy of this device, which is quite surprising if we take into account that this is one of the most important clinical problems for this type of patients. So we think, and one of the projects that we have conducted in our group is that there is the need to have some sort of top-down control, or providing a means to the patient for top-down controlling the lower limb muscles, while the robot is providing some type of assistance, whatever it is. In the case of the Locomat, it is robotic guidance. But what the company offers is basically a torque-based biofeedback, which is an average of the torques that the joints are observing based on the interaction of the user with the exoskeleton. So this is the type of top-down control that the commercially available system provides. So what we asked ourselves, is it possible to design different types of biofeedback, in particular neural feedback, and add it to this type of intervention with a well-known and established skeleton such as the Locomat? And our main question, or the major outcome of the clinical study that we conducted, was to observe if there were some improvements in spasticity. So this is the first question in this first part of the talk. So the bad news was that we actually conducted the trial and we didn't observe any significant changes in the performance of the users while they were doing the trials. But the good news is that we indeed observed an impact on the spasticity at the level of the 3 joints of the lower limb. So these are the 2 types of biofeedback. This is a screenshot of what the Locomat provides to the patients. Left hip, left knee, right hip, and right knee. And these are the average joint torques values per step. So here are, for instance, 5 steps, and you have the individual values. And what the patient has to achieve is to maintain this line as flat as possible, as flat as possible. So what we have produced in the Better project, funded by the European Commission, was a neurofeedback based on the activity of the lower limb muscles of the affected side of the patient, specifically vastus lateralis, rectus femoris, gastrocnemius, and soleus, within the gait cycle. So we tested different designs, different approaches, and we came with this kind of, crowded, somehow dense visual representation of biofeedback, but in the end it was manageable by the patient. So I will try to explain briefly. Here you can see the presentation of the biofeedback based on EMG on 1 gait cycle for a patient at the beginning of the clinical trial, and this is an intermediate stage. So, for instance, if we focus at the swing phase, you can see, for instance, that in red, the biofeedback is telling the patient that he or she is overactivating the extensor muscles if compared to a specific pattern that we define or the clinicians define. So this is the result of comparing the actual average EMG signals with respect to what should be a normal gait pattern in terms of EMG for the patient, and it would show an underactivation level with with blue. So the more overactivated the muscle at a specific subgait phase, the more red these boxes would become, the more blue, the more underactivated. So the ultimate goal for the patient should be to try to remove all the colors from these stripes. So for instance, here you can see an improvement in the swing phase. So the way that the patient is dealing with the ankle joint muscles in the swing phase, for instance, shows an improvement. Here he moved from an overactivation to an underactivation, and so on. So this was the type of biofeedback that we have shown to the patients, and we designed a randomized crossover clinical trial using the Locomat robot. We recruited 12 subacute non-ambulatory stroke patients that have problems to ambulate, of course. And we tested, as I mentioned before, in this project, different types of neural feedback. We also tested, by the way, EEG as a way to provide different types of feedback to the patients, but it was really not feasible from a methodological point of view, because the only stable signal was at the beta band, but it was a little bit too late for the effects of a top-down control, so we just stick to the EMGs that are underneath these trousers. So you have to believe me that the EMGs are there. And we created this condition in which we had two groups, and we only changed the biofeedback. So that was the only component of the treatment that was changed. So the patients underwent 12 training sessions with a constant guidance force at the 4 joints. So by the way, the Locomat exoskeleton doesn't have any actuators at the ankle joint level. So patients basically do not receive any kind of assistance to the plantar flexor or to the dorsiflexor muscles at the ankle. As I said, we have compared the effects of the two types of biofeedback. So as I previously mentioned, the disappointment was that we didn't see any immediate effects while the patients were using the system during the training with the two types of feedback. But the nice result is that we did found significant spasticity reduction in the hip, the knee, and the ankle joints. If we compare the EMG, which is this red posterior and in a lighter color like pink before the treatment. So you can see here the improvement, for instance, in the modified Ashworth scale of spasticity before and after the treatment with our top-down based neural feedback, which was not observed in the control group with the Locomat biofeedback. So this was one main result that we have recently published at GNR. Okay, so this was the first part to show you that we could use the biofeedback, different modalities, to try to enhance a specific or to induce specific neuromuscular contractions.
Can you explain why you have an effect on spasticity while the standard treatment would not? The control condition, which is the standard look. Do you have an insight in that? Why the difference?
Yeah, I would say that basically, if you provide only the overall net joint to the patient is not really understanding to what degree he has to put an effort on activating one muscle of a specific gait phase or the other. So we think that this is more specific to the condition. This is actually what the physiotherapists are normally doing when they treat the patients in the Locomat. They observe what the patient is doing and they are telling them, try to push forward, try to push harder, for instance, like with the whip. But this is like a type of feedback that is directly informing them about the neuromuscular control strategy, which is in the end what should explain somehow a change in the central nervous system that would lead to improving this hyperexcitability, this tone, or these abnormal reflexes that the physiotherapists normally measure with passive mobilization. So it's very speculative, but we think that is basically because it's more similar to what the manual physical therapy does.
So the patient literally sees this display?
Yes.
Okay.
Mhm.
It's very interesting, no? Because it means there's some cognitive root to affect spasticity.
Yeah, that's the point. We wanted to enhance the attention, and for that we also tried, as I mentioned before, this EEG-based system, but with all the electronics and the electricity and all the mechanics, the EEG was way contaminated for doing so. So that was the main goal of the project. And as secondary, we said, okay, let's rely on the neural signals at the periphery. But I certainly think that we should try to combine.
Are you measuring spasticity with a clinical scale or with?
Yeah, it's the modified Ashworth scale.
Ah, okay.
Mm-hmm.
So it's not during gait.
It's after. This is after. Yeah.
So would you say that if you measure spasticity during gaiting, it would be different, the measure, or you think it corresponds one to one? What do you think?
That's a good question, but I really don't know about a method to measure spasticity in dynamic, in volitional control of movement, because these scales, as far as I know, are passive mobilization.
But since you have recordings from the electromyography, maybe you could try to extract a measure of spasticity during.
Yeah, that's a very good idea. But we tried to see if there were changes in the EMG activation signals that were significantly different between the both types of feedback, and they were not, which was very very disappointing. But the good news is that the after effect seems to be positive. Yes.
Why is the feedback more effective at specific stages of the gait cycle and not others?
So the feedback is, as a whole, is providing information about the recruitment of flexors and extensors of the ankle and the knee. Okay, so the muscles take action depending on the subgait phase, and some patients may have more problems, for instance, in doing the plantar flexion appropriately, whilst other patients may have more problems in, for instance, flexing the knee during the last part of the swing phase, of the stance phase. So there are different features for specific patients. So there is some specificity of the patients that could be a compounding factor.
So there's no consistency across patients, it's just specific to.
So the tool is the same,
yeah,
but the outcome of the biofeedback might be different between patients. Because the patients in this group were subacute patients that were highly affected, and they had very different muscle force scales as well. I am not showing this, but it is included in the paper if you're interested.
Also, did you measure the spasticity just after the end of the training? Or
yes.
Just could it be a transient effect from higher effort in one of the groups during the training or something like that? Because maybe the biofeedback group, they are optimizing the cost of the gating pattern, or
we didn't look into it, but we did usability and satisfaction questionnaires, and in general, the patients were more satisfied by the neural biofeedback. So that could inform us about that, but we would have to look into the effort, the cost of walking, the metabolic cost, and so on. But that's a very good question. Okay, so I will move now to the second part of the presentation, which is about promoting motor learning to improve the control of the ankle muscles with a wearable robotic tool that we call Biomods. So we were interested in defining, using the challenge point theory as good as possible, a way to intervene with an ankle exoskeleton to induce motor learning in healthy firms, to try to see if there was some sort of correlation with the improvements in performance with respect to the cortical excitability of the motor areas, the muscles of the ankle joint. So sequence of thinking when designing this was that, okay, we wanted to have a video game that would provide the visual feedback, and we would present some physical challenge to the user by applying what we call haptic adaptive feedback with this control rule that is enabled by this compliant actuator. So there are different components in the system. One is the mechatronic solution, which is quite a nice system because it's inherently compliant, which means that you can backdrive very easily this ankle exoskeleton, as is not the case with the direct drive actuators with harmonic drive reductors and so on. So we designed this specifically for this treatment, and it has a compliant element that allow us to control the torque that is produced without the need of having a torque sensor, because we have a model of the compliant element and we can estimate what is the torque and so on. But in general, the idea is that we have also what we call a haptic adaptive feedback, which is based on a rule called tacit learning that has been proposed by Riken in Japan, in which they have demonstrated that if you have this accumulator just before the output of the PID to the motors of a biped robot, they have demonstrated that this could be a mechanism for induced learning tacitly. So you will not need to predefine any type of force patterns, but this type of accumulation would lead to guiding the motors. Yes.
So this is inducing errors into the PID controller, right?
This is basically inducing errors, but it can be an accumulation. Yeah. And our idea is to use this constant to regulate the error, as you were saying, as a function of the performance. So basically what this does is a reward system that is based on providing the autonomously customized task difficulty to the user to potentially promote learning. So in summary, the system is providing more effort if you are performing better. Okay, so the reward works in that fashion. So if you are a patient that, for instance, at a specific point of the treatment is not able to cope, the system will just reduce the amount of perturbation. So remember that we have the idea of having a trajectory, and what we want to apply is a perturbation. But we don't want to apply random perturbations because, as I mentioned, we would like to have some kind of give the patient the possibility of predict what is coming in terms of perturbation. So we are applying sort of a perturbation torque profile that can be either towards plantar flexion or towards dorsiflexion. And I will show you in a video how this works. So this is a trial in which the patient triggers the video game. These are resting position, and the exoskeleton is providing this perturbation profile, while the user should try to collect all the values to maintain this trajectory. So in the longitudinal study of motor learning induction with this, we had different types of profiles, of trajectory profiles, and different types of perturbation profiles. So in a way, the patient or the user knows the type of perturbation, but the amount, the magnitude of the perturbation is estimated based on the interactions at the level of the actuator. So that's why it's important to have this mechanically compliant exoskeleton, which is more resembling our muscles as actuators, which are inherently compliant. So this is the protocol that we have conducted with 10 healthy users, and I will explain a little bit more about this assessment with TMS in a few minutes. But what we did was to conduct 3 training days with 40 randomized trajectories per each day, and we had a pre-assessment with TMS before the first day, and we have a post-assessment after the third training day, and then we had another assessment after 30 minutes, and then a post-effect after 24 hours. long-term retention. So the instruction, as I mentioned and I showed before, for the patient or for the users in this case, was to try to find a strategy to compensate for these disturbances. So they didn't know that the magnitude of the disturbances was being modulated based on their performance. They just knew that they should collect the values as good as possible. And we have these different possible trajectory profiles so that they do not anticipate all the trajectory profiles trial by trial. And we tested both dorsiflexion and plantar flexion. So we had, on the one hand, metrics that we collected or we extracted based on the information coming from the robot. So the score is the more simple one, is the percentage of the collected values on the screen. But of course, you can have a perfect score, but really do very badly if you are being perturbed by the robot and you're not able to compensate for that. So we also created another metric based on the root mean square error that was calculated by subtracting the performed trajectory from an ideal linear path. So this would allow us to have two ways to measure the instantaneous performance by means of the score, which is used for the haptic adaptive feedback modulation, and we use this RMS to analyze overall how the patient is learning or not. Sorry. So what are the effects of this haptic adaptive feedback in the performance of the motor tasks? We have seen statistically significant improvements on a daily basis in the score, and we did see a trend, but it was not statistically significant in the RMS values after the treatment. But we did find a correlation between the two variables. I will now present the results for these robot metrics in the case study that we conducted with one stroke patient. We included this patient just as a usability case study. It is just a one-shot observation, but I mean one-shot is that we ran the full protocol. We even extended to 5 days, but we cannot conclude with regard to the functional scales or clinical status of the patient, because this was just a case study in which the patient was also receiving the traditional therapy in parallel to our treatment. But we did observe significant improvements in both main robot-based metrics, the score and the linear path, the RMS, after each day of training, which is very nice and is also coherent with what we have seen in healthy. So the patient did learn and was able to cope in general with the type of perturbations. I have to mention that we have to size the magnitude of the perturbation to the capacity of the patient, of course. Okay, so in summary, the patients were able and the users were able to modulate the perturbations. We had this strong correlation, and we had a significant improvement in the performance. So we can claim that we can induce motor learning because we have other markers for analyzing what is happening at the central nervous system. And I will go to that right now. So the corticospinal excitability was assessed by recording the motor evoke potentials elicited by the TMS. So we have this TMS in the lab, and we can induce this motor evoke potentials at the lower limb muscles. So the procedure is that first you have to select specific muscle. So you find the hot spot for this muscle that elicits the higher amplitude of the motor evoke potential. And then you, at the resting state, define the resting motor threshold for the specific user. So we put this chair in the picture to show that we are sure that the patient or the user is relaxed, so that there is no depolarization coming from the spinal cord. And what we are seeing, or our hypothesis, is that the treatment would induce an increase in the amplitude. Sorry, I have a sore throat. I will not
Would
be
you like
able
some water?
to sing tonight.
Would you like some water?
Yes, please, thank you. So, what we analyze is the peak-to-peak amplitude to see if there is a consistent effect in the excitability of this muscle as induced by the magnetic stimulation. So the procedure, as I said, we find the hot spot, dominant leg, and we observe not only the tibialis anterior, because it's the most widely used in electrophysiology research, but we also, of course, were interested in see what was contralaterally produced at the soleus muscle, and we used the rectus femoris as a control muscle that would initially not be involved in the motor task.
You can still dance, no?
Yes. Hope so. Thank you, Paul. So we included these three muscles in this electrophysiological assessment, and what we found is that we did have an increase in the corticospinal excitability after the longitudinal robotic training posterior to the treatment after 30 minutes, but here is, although there is a difference with respect to the pre-assessment, it is not significant, but it's an increase, and probably this is due to fatigue. We really cannot say. This is something that we observed in all the users, in all the healthy volunteers, but the most interesting is that you can see a significantly high increase after 1 day post-treatment. So we can claim that this treatment is, at least for these control users, able to induce these changes at the nervous system and could be related to the improvements in the motor performance.
Did you do the testing or follow-up, long-term follow-up?
No, this is one limitation of this. So this is a preliminary work that we are still conducting, and now we are in the process of conducting sort of longitudinal observation to see which are the long-term effects of this. But we think that this could be more interesting in the context of the use of this system at home. We are still trying to figure out the best way to do so. And we are transferring, by the way, this system to a company that is producing it as a compact, nice solution for doing that. But I think that we would like to concentrate more on having large samples of users wearing and treat and using the system as a training tool at home based.
Are all the patients responding positively to the treatment, or are some of them that don't respond at all? Because in some previous talks on motor rehabilitation, we were talking about patients that don't respond to the treatments, and one of the proposals was maybe these patients have a corticospinal injury, and that's why they are not responding.
Yeah, that's a very good question. So one point is that some patients may not respond to a specific robot treatment. The open question still is, shall we classify them between responders and non-responders? Is this an appropriate way to conduct a clinical study, or shall we include them all in the same group but differentiate them based on their functional status? There are different opinions on that, and I really don't have a clear idea about it. In our case, we tried to test the robot and the intervention with another patient, but it was not good for the patient. So he was not really able to respond to the perturbations because of his functional status. So we should find a way to identify which users could benefit from this system as a tool. But for the intact, for the healthy, everyone was able to use it. So we demonstrated the usability of the platform and also the TMS as a complementary measurement that would be relatively easy to use before and after an intervention. So since we were not also able to conduct the TMS-based analysis with the patient because of the risk of seizures and so on. We conducted another type of analysis of the range of movement. So the range of movement evaluation consisted on moving up and down a ball on the screen. So this was after the intervention with the compliant exoskeleton. So the patient had to move the ball up and down, and there was an increase in the range of movement based on the performance or the achieved range of movement. So the patient, the lines, the horizontal lines changed to the maximum reached in order to make the task more difficult, but at some point these were being separated more and more, but the patient was not really aware of it. So this was the way that we evaluated the increase in the range of movement after each training day, and we also calculated the increase in joint velocity. So what we have found is that at the beginning, after the second day, there was actually a decrease in the range of movement. We cannot explain why, but there was a consistent increase throughout the days. While for the velocity, there was a sort of upward zigzag shape that could be explained by the changes in the range of movement, but in general, there was a trend towards increasing the joint velocity, which is a very important marker also for the spasticity issue, and this was also not correlated, but in line with some functional tests that we conducted with this patient. For instance, the time 10-meter walk test improved after, the 6 meters walking test also improved, the step test, the repetitions, which are stepping on stairs, almost doubled, and the time up and go decreased, which means an improvement in a few seconds. So in general, the robotic system was also an interesting tool for evaluating the range of movement and the joint velocity, and we could theoretically use these metrics to correlate them to the functional status of the patient. So in summary, we could conclude with regard to the electrophysiological measurements that we did find a significant increase in the tibial anterior cortical spinal excitability. We didn't observe this affecting the soleus muscle, and either in the rectus femoris. This is something that we expected because the rectus femoris, although is a biarticular muscle, is not involved in the task. But for the soleus muscle, we could rethink the way that we are doing the hotspot identification and maybe focus on the soleus muscle and expect a contralateral effect in the tibialis anterior. And as I mentioned, we had also a satisfaction questionnaire in these experiments, both with the healthy and with the patient case study. We found this net increase in the range of movement, and we also had a good acceptance. So I am going to conclude this second part and almost to finalize the talk and be prepared for some discussions. So we have proposed an integrated tool to elicit improvements in the performance of the user while doing a motor task to induce or enhance a motor skill, targeting a specific muscle function. This is a potential rehabilitation tool that we want to try to bring into different types of scenarios and probably could be useful for other types of patients with upper motor neuron syndromes. So we demonstrated the feasibility, and what is from the robotic point of view very interesting for us is that we have demonstrated a stable performance of this autonomous modulation of a haptic reward with this Biomod actuator. So some more general concluding remarks that I would like to give before I close the talk. So some recent studies point out that an increase in the cortical spinal excitability may be related to an improvement in motor learning. But extrapolating these findings on the cortical spinal excitability to learning processes that should take place in the recovery of a stroke is not so easy. So we should not try to reach that kind of conclusions by simply correlating these results. We should try to observe more in detail what is happening. But in general, the relationship between the improvements in the metrics of performance, the motor learning observations, and the measures of corticospinal excitability could explain some improvements in motor learning as a whole. And the last conclusion is that the smart adaptive controllers for robotic gait trainers are likely to be the way to solve the limitations of the current exoskeletons as tools for recovery of the stroke, but also spinal cord, Parkinson, multiple sclerosis, and so on. So one important point is to enhance the intelligence of these machines, but also to provide means for the patients to understand what they are doing while they are being treated by these systems. And with this last slide, I would like to acknowledge the work of my collaborators at Cajal Institute. The Biomods and collaborator is mostly the thesis of Guillermo Asimprieto, is about to read his dissertation, and also our collaborators in the Better and Biomods projects funded by the European Commission. Thank you.